The dengue virus is one of the flaviviruses, has 4 serotypes, and they generally being described as breakbone fever. The victims sometimes being quoted, they rather die instead of having a second time. And dengue can induce hemorrhagic fever, shock, encephalitis, and very often, not very often, but in a severe case, and also can cause death. There are 4 different serotypes, so people can be infected with one serotype and induce particular type specific lifelong immunity, but not actually immune to other types. So people, each one can get 4 times dengue infection, and often with increased severity. So it can be extremely debilitating to travelers if we bring back to this country, because we work with the Department of Defense. There's major concern, but our military sent overseas and potentially can be infected, and actually many of them have been infected and suffer through the infection. So dengue actually is a third world, considered initially third world disease. About 2/3 world population live in dengue-infested area, and there is about 100 million cases, but significant amount of the patient actually have to be hospitalized, and really patient have to suffer through for weeks, high fever, body ache, and encephalitis, and there's many diseases. But recently, actually, this disease has been spreading very close to the United States, and spread already in the Caribbean and Mexico, and actually one of the territory, Puerto Rico, actually NIH had a meeting a few years ago while there was having severe dengue outbreaks. There's many actually severe cases in Puerto Rico. So actually, because it's part of the United States now and increased travel, so there is a potential that dengue can be brought into this country. Actually, already in Key West, in Florida, and part of Texas. Now, recently we heard the news, and there's a worse West Nile outbreak again in Texas, and actually all over the United States. So there is a major concern in developing vaccines against the 4 types of dengue viruses. But why dengue virus has been, people say, last 60 years, people have been trying to develop dengue vaccines, and there has not been an effective, safe vaccine. There are several challenges, and actually one is you have to induce strong neutralizing immune response. So without neutralizing immune response, the immune response actually enhances the disease. We'll hear over again. And then secondly, you have to induce balanced immune response, antigen presentation among the 4 serotypes. And the underrepresented immune response not only will not be able to protect the disease, there is a concern it can cause immune enhancement of the disease, similar to the natural infection. When there's immunity against different serotypes, it's not sufficient. Actually, the antibody binds to the viruses and induce uptake of the virus and cause the more severe disease and spreading the virus. And we also need to improve the safety profile. The early vaccine technology, which is based on attenuated viruses, based on natural selection. So the natural selection of attenuated viruses is never a one-way street. So they can come back, follow the same path, or from alternative paths, actually become revertant, become equally virulent, or potentially more virulent viruses. So to resolve the challenge in dengue vaccine development, you have to present natural conformation of the antigen, the envelope protein, which is called E antigen, and in the type-specific manner. You have to induce neutralizing immune response against all 4 types, the envelope protein, and therefore actually require a balanced immune response, as is heard earlier. We cannot have an immune response, have a strong immune response against one type, and actually very weak immune response against another. Not only will not be able to completely protect against those 4 serotypes, but actually in some cases has the potential not cause diseases. So the importance is naturally expressing the glycoprotein. why in a natural infection we can have lifelong immunity against the virus. That indicates that you can induce lifelong immunity against the specific serotypes. But that immunity does not prevent the different serotypes infection. In many cases, actually induce an immune enhancement. So therefore, the one important issue is the glycoprotein. The amyloid protein has been naturally conformed, has been fully glycosylated, have properly folded. If you make a protein in E. coli, it's folded differently. And even if it's folded correctly after purification, the denaturing and renaturing process never complete. So not necessarily containing the receptor binding site. By definition, that's a neutralizing epitope. So you're presenting 4 serotypes, and they have to induce neutralizing immune response, not the cross-reactivity. Antibody binds to the envelope, not neutralizing it. Actually, through the Fc receptor, actually enhance uptake of the glycoprotein. So you have to have antibody bind to the neutralized epitopes and block the virus infection, therefore actually prevent the virus infection. So one of the immune enhancement mechanisms potentially, because you enhance the uptake of the virus, and the virus not be able to be neutralized and infect new host cells. And then once the CTL response, innate immune response, all mounting up against infected cells, you cause massive damage for the host cells. That was why you see most severe hemorrhagic fever, encephalitis, and the disease getting enhanced. So it is critically important you have to have naturally conformed the glycoprotein that presented receptor binding site. This is the simplified diagram of the amyloid protein. It's composed of the pre-M, M, and E protein. So actually the protein is conformed. First of all, there's natural conformations. It has to be maintained, formed during the virus budding and during the infection. They're being described as a mature form and immature form. Actually, the mature form, immature form actually transition depends on the condition, even after the virus budding, even our virus already being so-called mature. So the natural conformation is critically important. Certain stage, not only the glycoprotein not inducing neutralizing immune response, but worst of all, could actually induce cross-immune response that enhance the disease. So what type, how we develop vaccine? So our strategy is a sheep in the clothing of wolf. So we would like to naturally present the entire repertoire of the antigens, but inside is not a tenured virus. So it's a benign carrier. So therefore will not never cause disease. So the broader application of this technology, we can always say now we can put a sheep in the clothing of a wolf. We can put a sheep in the clothing of a tiger. We can put a sheep in the clothing of a lion. By transforming different highly pathogenic viruses based on what the insert we put into this carrier, and we can present a different vaccines. So attenuated viruses, for many of the so-called low-hanging fruit has already been picked. The reason is if the virus is being attenuated, that can be safe enough and can still cause infection, therefore can be an effective vaccine. But you can basically, it's a crippled wolf. You can't cripple a wolf, and we may win. If you cripple a tiger, the chance of winning will be less. You cripple a lion, you cripple an elephant. So it depends on the viruses we want to make it attenuate. That potentially can be, there's a limit what type of attenuated virus you can use. And one example I'm going to present later is the Ebola viruses. So basically we use Catabax, a complex endoviral vector system to expressing multiple antigens. I'm not trying to oversell the system because just basically an example. Any vectors you can put multiple inserts and to avoid to deliver the antigens inside cells. Actually, the antigen will be presented by a complex endoviral vector with multiple antigens. For in the case of HIV, we're not only expressing the glycoproteins, we can also express the capsid protein, GAP protein. So we can induce the naturally conformed glycoprotein will form on the cells exactly identical as HIV infection or on the surface HIV particle. This is important to induce neutralizing immune response, avoid the antibody actually not necessarily neutralizing. So for HIV viruses, the failed trial in the end is clear. So non-steroid immunity hardly can prevent any virus infection, certainly not HIV. So actually, that's which from the beginning we believe the HIV viruses and many viruses you need to have neutralizing immune response, and certainly you have to have a CTL response. The importance, the cell surface expression, it has all the co-receptors and actually enhance immunity in a natural way as a natural infection. So in the case of HIV, we can not only causing the vector to produce the glycoprotein, we can produce gag protein. They form natural empty viral particles because there's no HIV genome. So the virus particle will be empty and then mimicking natural infection. And in the case of HIV, because it is a chronic infectious disease, have immunosuppressive function, in the complex vector, we also expressing cytokines, actually to stimulate immune response, mimicking the natural helper cells. So basically it's a concept. You have expressing multiple antigens, that antigen epitopes that responds to neutralizing immune response, CTL response, and in the case of chronic infection, you can potentially enhance immunity by locally in situ expression of the cytokines. So it's important to expressing the cytokines as where the immune response starts, not as systematically actually potential causing side effects. So the vaccine actually is the concept is expressing multiple antigens and also potentially enhance the cytokines, immune enhancement to combat chronic infectious viruses that often has the immune suppression function. And what actually remodify the full spectrum immune response not only has to have antibody response, CTL response, certainly innate immune response will join, but also have multiple response to multiple antigens, epitopes that are crossly neutralizing or protect against a broad range of subtypes for a virus infection. So as mentioned, so some of the viruses are highly lethal. One of the good examples is Ebola viruses, Marburg viruses. We heard on the news the recent outbreak in Kenya. Actually, there's a high level of mortality, and potentially it can be transmitted by just contact with food. And actually, recently recognized, not necessarily you have to kill chimpanzee or touch that meat. By shaking hands, the natural contact, as long as you have transmitting the body fluid that contains the viruses, you potentially can be infected. And also recently, the military has been doing the research against biological weapons, shown it can be aerosolized. And first infection is in the lung, and the early symptoms is coughing. And also there's high titers of virus. It's not natural pathway of infection, but once you introduce in the lung, the early symptoms is coughing. Has very high titer viruses, and in the non-human primates model, a single virus inhalation or infectious particle can cause nearly 100% death. And again, fatal virus has multiple subtypes, depending on how you classify it. There's 9 different types of Ebola Marburg viruses. So it's difficult to protect not only one type of viruses, it's even more difficult to protect against all 9 serotypes, and there is 3 types of Ebola, multi-type Marburg viruses. So the multiple form, the multi-antigen expression, we can use a single vector expressing multiple antigens. In this case, is Marburg virus Mazuki, Marburg virus CX67, Ebola virus Zaire, and Marburg virus Rabbit. Basically, a single vector expressing the glycoproteins of 4 fetal viruses. And this is the feces staining with specific antibody. You can see on the surface the cells the high-level glycoprotein being expressed, most of the naturally infected cells, because naturally infected cells have a cytotoxicity. So the cells will not be efficiently expressing the antigen of interest. In this case, you're basically delivering a vector. The vector is expressing the glycoproteins on the cell surface. So what is the sheath in the closing wall? So these are the sheaths in the closing wall. So the cells are expressing high-level glycoprotein on the cell surface. In this case, with a single vector transduction, we can expressing three types of Marburg viruses, one type of Ebola viruses. Actually, we increase the technology, increase the capability with a single vector expressing the glycoprotein of multiple types. More importantly, the glycoprotein on the cell surface are naturally conformed the same way as the virus infection synthesized its amyloid protein. So the system is quite efficient. As I said, this is actually not staining the viral protein, but a GIP. When we put a GIP, inject in the muscle, you can see under the second scope the entire muscle lighten up. So if these are the antigens of HIV, or antigens of Ebola, antigens of dengue, so that was causing the immune response, as if these muscle cells being infected by the virus. We all know HIV do not infect muscle cells, do not express the protein at a certain level. But if we can use a vector expressing HIV glycoprotein in the muscle in such a high level, and especially we actually increase local expression cytokines and gag protein-induced CTL response, we can expect that the full spectrum immune response against HIV, as if there is infection taking place. So, in the case of a Marburg Ebola, and by itself actually is highly important to develop vaccines against Marburg Ebola, not only prevent natural infection. And there is more tourists going to Africa, potentially bring the disease to a civilized, more urban environment. If you get into a large city, actually have Ebola outbreak, Marburg outbreak, and the potential is going to be disastrous. Certainly is a biological weapon potential threat or military. So this by itself is highly important to develop vaccine against Ebola Marburg viruses. In addition, we believe it is one of the best models to testing if a vaccine platform works or not, because Ebola virus and Marburg virus infect any primates as far as we know, including humans. So actually, non-human primates is not a model. They naturally infected by Ebola Marburg virus. The chimpanzee, any great apes and monkeys will infect by Ebola Marburg virus, like humans. The disease process, like everything, is indistinguishable. The monkey will die within days, and actually suffering the same hemorrhagic fever symptoms, and every pathology is almost identical to human. So with one vaccination in this case actually is multivalent. This is one challenging experiment, and out of many serotypes, we actually done by U.S. Army, led by a group, Bill Pratt. So with a single vaccination, you can see the antibody goes up to very high level, maintained at very high level at 84 days. And at about 160 days, the non-human primates were challenged with 1,000 times the lethal dose of the virus. Not the lethal dose, 1,000 times the lethal dose by the military standard. And all vaccinated animals survived. None of them, of course, they controlled that. And then we look into the antibody response. You can see the antibody response remained the same level as the post-vaccination. So it indicates this vaccine increase already reached the maximum level plateau. And even after infection, the antibody level could not further increase. Then the same group of monkey being challenged again with Marburg, first time the Ebola virus, second time with the Marburg viruses, again with the 2000 LD50, about 1,000 times lethal dose. Again, 100% survival, and then you look into antibody against Marburg viruses. Again, is the after post challenge, this viruses actually the non-human primates generate the same level of antibody. Again, further indicate the vaccine actually protects all the non-human primates. So how actually we're supposedly talking about the dengue viruses, and again, how we're going to develop dengue viruses. Dengue virus is not as lethal as Ebola Marburg viruses. There are 4 serotypes so far. So we believe it's less of a challenge, maybe a too large a claim, but less of a challenge compared to Marburg Ebola viruses. But actually, Marburg Ebola viruses, there's 9 types we have to protect. Dengue, there are 4 types. As I mentioned earlier, we need to induce a balanced immune response and present 4 serotypes naturally in an identical level as if in an ideal situation. So we created the first experiment to create 2 vectors, each one expressed by a CMV promoter controlled cassette. So if you can look into the 4 serotypes expressed at an identical location, identical cassette, so we were hoping that will generate identical level antigen expression inside the cells. So therefore, ideally, generate identical immune response. Therefore, we can present the natural tetravalent immune response. And importantly, this vector is not replicating. The envelope protein have nothing to do with the vector replication. Therefore, will not affect the level of antigen expression, except the expression by the cassette. So in this case, the CMV promoter to achieve high-level expression. So again, the similar experiment was done by NMRC, led group by Kevin Porter. They did a challenge in 2 sets of non-human primates. A large number of non-human primates was vaccinated, and for dengue, because initially when you have a 4-type immune response, cross immunity can provide some level of neutralization. So it's only when the initial early acute immune response wanes, and then actually the cross immunity can cause enhancement. So therefore, actually, based on the NMRC suggestion, there are 2 groups of monkeys. One is vaccinated, challenged at 12 weeks. The second group challenged at 24 weeks. So actually look at the immune response and look at the challenge result. So far, actually, there's no real live and death in non-human primates models, and the non-human primates do not exhibit the dengue symptoms. But actually, the best way to look at it is one is the immune response, and secondly, the neutralizing titer, and secondly is the viremia. So this actually neutralizing titer, the point is to see the highest is 1400 and lowest a few hundred. And it's hard to read, but actually, for the bar representation, you can see the scale is wrong. But the proportion is correct. After 29 days, you can see the immune response against 4 serotypes that increased with the time by 85 days, similar to Ebola Marburg viruses, which maximum level. But important to see is, in this case, there's differences in DEN 1, DEN 2, DEN 3, DEN 4, and actually the DEN 4 being the lowest, DEN 2 is the second lowest, but DEN 3, I like to point out, in general, DEN 3 is lowest immune response, cause most of the disease. In this case, and I'll mention later, we see the highest level against DEN 3 because it's not related to virus replication, but related to gene expression. And this is days of viremia. Actually, what we can look at in the control, the 5-7 days viremia in the vaccinated group, and the maximum is 1 day, close to 1 day viremia in one of the non-human primates. And actually, that's DEN 4. DEN 2 has 0.3 days of viremia in the study. Then also we're looking at 2 groups. And the first 12 weeks challenge, basically we do not see any, the orange bar is the control. We do not see viremia in DEN 1, DEN 3, and we see slightly viremia in DEN 2, and a slight viremia in DEN 4. After the 24 weeks, we see a similar picture, but actually there's over 2 days viremia in DEN 4. So this shows complete protection against all 4 serotypes, but there is so-called slightly breakthrough in DEN 4. If you look at the immune response, that's the top graph. Actually, DEN 4 has the lowest immune response. For whatever the reason, even you're expressing at the same promoter, Actually, DEN 4 is lowest, DEN 2 is second lowest, have slightly viremia, indicating if the immune response goes to a higher level, you can completely block the viremia in this case. Sorry. All right. So actually, what we done in DEN 3 is not only is have the lowest immunogenicity, but it is very difficult to clone it in the plasmid DNA. So what we've done in DEN 3, we humanize the genome, the codons, by modified DNA sequence. Therefore, actually, we can better clone it, better express it. So based on this, we made the lowest immunogenicity become the highest. So actually, therefore, we actually humanize all 4 serotypes, and after humanization, you can see all 4 serotypes induce same log, same level of immune response. With a single vaccination, goes to maximum level maintained for a long period of time, and the immune response now actually you can see is much more balanced compared to previously. So actually, with DEN 1, DEN 2, DEN 3, DEN 4 being at almost equal level, and based on this immune response, actually we can achieve complete protection against all 4 serotypes, not even the shred, slightly viremia. All right. Okay. And with that, I will basically say this vaccine platform can be administered from nasal spray, from skin patch, and we actually have an oral capsule. So this is some of the data to show expression. I would like to thanks. We built a facility to housing the production. I'd like to thank the NIAID provide most of funding, Navy and U.S. Army provide a challenging model.